Hello everyone, and welcome to our session today. I'm excited to introduce you to Module 1, General Introduction to Medical Biochemistry. Today, we'll be focusing on Study Session 3, which covers the functions of the cell membrane and the organelles. This is part of BCHM 241, the biochemistry of macromolecules. Over the course of this presentation, we'll explore how these cellular structures work together to sustain life at the molecular level. So, let's get started and dive into the fascinating world of cell biology and biochemistry. The cytoskeleton has many functions that are essential for the cell's overall health and activity. It provides support and gives the cell its shape, acting like a framework that keeps everything in place. It also helps position and transport organelles within the cell, ensuring that everything is where it needs to be for the cell to function properly. Additionally, the cytoskeleton provides strength, making the cell more resistant to mechanical stress. It plays a crucial role during cell division, helping to organize and separate chromosomes accurately. And of course, it aids in cell movement, allowing cells to crawl, change shape, or even move materials around. All these functions highlight just how vital the cytoskeleton is for maintaining the cell's structure and enabling its dynamic activities. Let's take a closer look at the functions of the endoplasmic reticulum, starting with the smooth ER. The smooth ER is involved in several critical processes, including xenobiotic metabolism, which is how our body detoxifies foreign substances. It also plays a key role in releasing calcium ions, essential for various cellular activities, and in lipid synthesis, which is vital for building cell membranes and producing signaling molecules. Moving on to the ribosome, this is where ribonuclear protein synthesis occurs in the nucleus. Ribosomes are the cell's protein factories, translating genetic information into functional proteins. Now, the rough ER is closely associated with protein synthesis. It provides a platform for assembling proteins that are destined for secretion or for use in the cell membrane. It also manages protein movement within the cell, ensuring proteins reach their correct locations. Additionally, the rough ER is involved in proofreading, which helps maintain the quality of proteins being produced, preventing defective ones from accumulating. Finally, when we talk about ribosomes and the endoplasmic reticulum together, it highlights how these structures work seamlessly to support the cell's overall function, producing, folding, and distributing proteins efficiently, which is fundamental to cell health and activity. Let's talk about the lysosome, which is actually not found in plant cells. Its main job is to help digest food or cellular invaders like bacteria and viruses. Think of it as the cell's recycling center and cleanup crew. It breaks down old or damaged parts of the cell so they can be reused or disposed of properly. Another really interesting function is its role in cell suicide, or apoptosis. While that might sound a bit harsh, it's actually a good thing. It helps keep the overall health of the organism by removing cells that are no longer functioning properly or are potentially harmful. So, in essence, lysosomes are vital for maintaining cellular health, but it's important to remember they're absent in plant cells, which use different mechanisms for similar processes. This highlights how different cell types have unique ways of taking care of their internal functions. So here we see a macrophage, which is a type of immune cell, actively fighting off potential threats. It has ingested a potentially harmful bacterium through a process called phagocytosis. You can imagine the macrophage wrapping around the bacterium and forming a vesicle, which is like a little bubble that contains the pathogen. Now, for the next step, this vesicle doesn't just sit there. It fuses with a lysosome inside the cell. Lysosomes are specialized organelles filled with enzymes that can break down and destroy pathogens. When the vesicle merges with the lysosome, the enzymes inside can then act on the bacteria, effectively destroying it and preventing infection. It's also worth mentioning that this process is just one part of the immune response. The cell has other organelles working behind the scenes, but for simplicity, they're not shown here. The key takeaway is how lysosomes play a critical role in pathogen destruction, which highlights the importance of cellular defenses in our overall immune system. Mitochondria, as you might already know, is the plural form of mitochondrion. These tiny structures are found in both plant and animal cells, and they play a crucial role in the cell's energy production. Think of the mitochondrion as the powerhouse of the cell because it's where most of the cell's energy is generated. Without mitochondria, cells wouldn't have the energy they need to perform all their functions. So, understanding mitochondria helps us see how cells stay alive and active, supporting the overall health of plants and animals alike. All right, let's dive into the structure of the Golgi apparatus. Imagine it as a series of flattened, disc-shaped sacs or vesicles stacked together. These sacs have two distinct faces that are key to their function. The first is the trans face, which is essentially the receiving side. It's oriented toward the endoplasmic reticulum and the nucleus, receiving proteins and lipids from these regions. The other side is the cis face, which acts as the releasing face, oriented toward the cell's plasma membrane, where the Golgi dispatches processed molecules. Now, what does the Golgi do? Its main role is to store, modify, and package proteins and lipids. It's like a cellular post office, making sure these molecules are properly processed and sent to their next destinations. The transport between the Golgi and other parts of the cell happens via vesicles, tiny membrane-bound sacs that ferry molecules back and forth. So, in essence, the Golgi apparatus is crucial for maintaining the cell's internal logistics, ensuring that proteins and lipids reach their correct locations in a well-organized manner. Let's take a closer look at the boundary of the cell. This boundary is made up of a phospholipid bilayer, which is essentially a double layer of phospholipids. This structure is crucial because it acts as a barrier, controlling what enters and exits the cell. We often refer to this boundary as the cell membrane. The cell membrane isn't just a simple barrier. It's a dynamic and flexible structure that supports the cell's environment and helps maintain homeostasis. Understanding this membrane is key to grasping how cells interact with their surroundings and how they function as living units within the body. Now, let's dive into the different types of transport across the cell membrane. First, there's passive transport, which doesn't require energy. Under passive transport, we have simple diffusion, where molecules move directly through the membrane from an area of high concentration to low concentration. It's a straightforward process driven by the concentration gradient. Then there's facilitated diffusion, which helps larger or polar molecules cross the membrane using specific transport proteins. This includes uniporters, which move one substance in one direction, antiporters, which exchange one substance for another in opposite directions, and symporters, which move two substances in the same direction. Moving on to active transport, this process does require energy, usually in the form of ATP. Primary active transport directly uses ATP to move substances against their concentration gradient. An example is the sodium-potassium pump. Secondary active transport, on the other hand, relies on the energy stored in electrochemical gradients created by primary active transport to move other substances. We also have bulk transport mechanisms, which involve large quantities of materials. Exocytosis is a process where cells export materials by enclosing them in vesicles that fuse with the membrane. Endocytosis is the reverse, where cells internalize substances by engulfing them in vesicles. Both of these processes are essential for transporting large molecules or bulk quantities across the membrane. Throughout this slide, the key idea is that transport across the membrane is vital for maintaining homeostasis and allowing cells to interact with their environment efficiently. Whether passive or active, each mechanism plays a unique role in cellular function. Moving big particles or fragments of cells is called bulk transport. This process allows the cell to move large objects across its membrane. One way this happens is through endocytosis, which is when materials are brought into the cell. Within endocytosis, there are different types. Phagocytosis, often called cell eating, involves taking in big molecules or particles. Then there's pinocytosis, which is when the cell takes in large amounts of liquid. Another specialized form is receptor-mediated endocytosis, where only specific molecules that match receptors in the membrane are allowed inside. This selectivity is crucial for the cell to control what enters. On the flip side, exocytosis is the process of taking materials out of the cell. Both endocytosis and exocytosis are essential for maintaining the cell's environment and communication with its surroundings. These processes are fundamental to how cells actively manage their internal and external environments, supporting their overall function and health. Now let's look at the three main variations of endocytosis. First, we have phagocytosis, which is often called cell eating. In this process, the cell membrane wraps around a large particle, like a bacteria or debris, and then pinches off to form what's called an intracellular vacuole. This allows the cell to engulf and digest large particles. Next, there's pinocytosis, sometimes known as cell drinking. Here, the cell membrane engulfs a small volume of fluid along with any dissolved substances. The membrane pinches off, creating a small vesicle inside the cell. This process is important for sampling the extracellular fluid and nutrients. Finally, we have receptor-mediated endocytosis. This is a more targeted form of endocytosis. Specific substances bind to receptors on the outside of the cell membrane. Once the substance binds, the membrane forms a vesicle around it, bringing in only that particular molecule or set of molecules. This process is highly selective and efficient, allowing cells to uptake specific nutrients or signaling molecules. Remember, all these forms of endocytosis are essential for the cell's ability to interact with and adapt to its environment, and they help maintain cellular homeostasis. In exocytosis, a vesicle actually moves toward the plasma membrane, which is the outer surface of the cell. Once it reaches the membrane, it binds to it, creating a connection point. Then, the vesicle releases its contents to the outside of the cell, effectively exporting materials like waste, proteins, or signaling molecules. This process is essential for many cellular functions, including communication and maintaining the cell's environment. And remember, this entire process is a highly coordinated way for cells to interact with their surroundings, supporting the overall theme of cellular transport mechanisms. The image credit here is a modification of work by Mariana Ruiz Villarreal. Let's take a closer look at some key organelles and the biochemical reactions that happen within them. First, the mitochondria, often called the powerhouses of the cell, are responsible for cellular respiration, producing ATP, which is the main energy currency. Next, the nucleus serves as the control center, where DNA replication and transcription occur, allowing the cell to produce proteins and replicate itself. Lastly, the endoplasmic reticulum, especially the rough ER, plays a crucial role in protein synthesis and folding, while the smooth ER is involved in lipid metabolism and detoxification. Now, shifting focus to lysozymes, these are enzymes that break down bacterial cell walls. You find them prominently in tears, which is fascinating because tears aren't just for lubrication. They also serve a protective function. The lysozymes in tears help defend the eyes against bacterial infections by destroying bacteria that come into contact with the eye surface. Essentially, tears act as a first line of immune defense, and lysozymes are a vital component of that system. Their presence in tears underscores how our body integrates multiple defense mechanisms to keep our eyes healthy and functioning properly.